Alzheimer's disease by Maria Tully, Lucy Specht, and Emily Parry.
So in order to understand how Alzheimer's disease develops in the body, we must first understand how cells communicate. First, there are two types of cell signaling, local signaling and long-distance signaling. Local signaling occurs when cells communicate via direct contact between membrane-bound molecules with cell-to-cell recognition. And then long-distance signaling occurs when organisms secrete hormones that travel throughout the body and signal different responses.
Cell signaling can be divided into 3 steps. 1. Reception. Reception is the binding of the signaling molecule to the cell's receptor protein, which triggers the detection of the molecule. Transduction is a series of steps that converts a signal to a form that brings out a specific cellular response, usually requiring a signaling transduction pathway. 3. Response is the activation that is triggered by the signal.
There are 2 types of issues within neurons that are associated with the cause of Alzheimer's. To take a look at plaques, we will begin in the cell membrane. Normally, the amyloid precursor protein, APP, which helps repair neurons, is cut up by both alpha and gamma proteins. This causes a soluble molecule to be formed, which will go away in the extracellular fluid. However, in some cases, another enzyme, beta secretase, teams up with gamma secretase and causes an insoluble molecule to be formed. This is the monomer amyloid beta, which we call plaque. Clumps of amyloid beta monomers stick together, which can get in between neurons and potentially disrupt neuron signals. For tangles, we must look inside the cell itself. Neurons are held together by various microtubules. The protein tau ensures that these microtubules do not break apart. The buildup of beta amyloid, or plaque, on the outside of the cell initiates the pathways on the inside of the cell. This then leads to the activation of protein kinase, an enzyme that transfers phosphate groups to proteins, in this case tau. Protein kinase causes tau to change shape, stop supporting microtubules, and clump up with other tau. Neurons with tangles and non-functioning molecules cannot signal as well, leading to characteristic findings of Alzheimer's.
Less than 10% of all people with Alzheimer's have early onset Alzheimer's disease. When people do have it, it occurs in ages 30 to 60. It is caused by an inherited change in one of 3 genes, resulting in FAD, familial Alzheimer's disease. This involves single gene mutations on chromosomes 21, 14, and 1. 21 is an abnormal APP, 14 is an abnormal presenilin 1, and 1 is an abnormal presenilin 2.
When people have late onset Alzheimer's disease, their symptoms become apparent in their mid-60s or later. A genetic risk factor for late onset Alzheimer's is having a form of the apolipoprotein E gene on chromosome 19, which is right there. APOE comes in several different forms. APOE2 is rare and it may provide protection against the disease. And if the disease occurs in a person with this allele, it develops later than in people with the E4 gene. And APOE3 is the most common allele, and it doesn't increase or decrease the risk of the disease. APOE4, however, increases the risk of Alzheimer's disease, and it's associated with earlier age of disease onset. So basically, if a person has more APOE4 alleles, they have a higher risk of developing the disease. And remember, these are just risk factors. So if a person has one of these, they can do anything they can to prevent it, and it's not certain that they're going to get the disease.
Symptoms of Alzheimer's include trouble with memory, thinking and reasoning, making judgments and decisions, planning and performing familiar tasks, and changes in personality and behavior. Alzheimer's may also cause parts of the brain to physically shrink or swell.
If you have any other questions about Alzheimer's or cell signaling, you can refer to the sources below.